latest work.
Thank you so much. Thanks so much to the organizers for giving the opportunity to share this. So it's a project that I actually started during the tail end of my postdoc at Stanford, and I've been working on it with some people from Howard Chang's lab at Stanford. And now I've just started my own lab at McGill just 6 months ago, and so my lab is sort of trying to take over some of the work here. So we're really interested in protein-RNA interactions and RNA-RNA interactions. And so we're investigating the role of a protein called poly-RC binding protein 2 in the HCV life cycle. So first, what are poly-RC binding proteins? They're single-stranded RNA binding proteins, and really they're the major cellular RNA binding proteins in the cell. They are typically characterized by their 3 KH domains, which are RNA binding domains. And they preferentially bind to poly-C sequences, but they're also known to bind to poly-Us. They typically bind to protein coding genes, and they interact usually in the 3' UTR of the genes from what is known about PCBP activity. There's 2 main isoforms in mammals, and they can be homo or heterodimeric with other HNRNP proteins. And PCBP2 is interesting because it's actually more recently been shown to be induced by interferon and also by viral infection. And the PCBPs in general, their RNA binding activity leads to effects on mRNA processing, RNA stability, and translational control. So I got a bit interested in PCBPs because they actually have a history with viruses. And so the most well-characterized of these is actually in poliovirus. And RNA viruses have a bit of a problem. So they have to both translate their RNA and replicate their RNA. And a polymerase can't travel along the RNA while it's actually translating. So the ribosomes and the polymerase can't happen at once. There has to be some kind of mechanism to switch off translation and initiate RNA replication. And so in poliovirus, PCBP is actually this switch. So it binds to the 5' end of the poliovirus genome. It also binds to the viral iris. And this binding actually helps load ribosomes onto the viral RNA so that you actually get translation of the viral proteins. Now, once you get enough translation of the viral proteins, one of the viral proteins called 3CD is a protease, and it actually comes and it cleaves the poly-RC binding protein. And this means that the poly-RC binding protein can now no longer load ribosomes onto the RNA. The RNA is cleared of translocating ribosomes, and now the PCBP that binds to the 5' end of the genome can actually help circularize the genome and initiate negative strand RNA synthesis. So it's a really elegant way that the virus has taken over this host RNA binding protein to mediate this switch from translation to replication. And it's also been implicated in the life cycle of other viruses like rabies, VSV, or HPV. Finally, a recent study also told us that it is involved in innate immune responses. It's actually a negative regulator of MABs-mediated antiviral signaling. So we were actually curious a little bit about if this might be involved in HCV replication or the switch. And so I looked into the literature, and we found that PCB depletion actually does reduce HCV RNA proteins and RNA and proteins, but it's unclear whether the PCBs are able to affect translation or replication or both. It interacts with the HCV NS5A protein, which is a core component of the replication complex. And it's been suggested and sort of shown to bind to both the 5' and 3' UTR of the virus, specifically to stem loop 1 of the virus. And an unknown region in the 3' UTR, and it's speculated to have some kind of role in circularization. So we wanted to know what really is this role of this protein in the HCV life cycle. Is it circularization of the genome? Does it mediate this switch from translation to replication? So as some of you will know, one of my main interests is actually in an interaction at the 5' end of the genome with a human liver-specific, highly abundant microRNA called microRNA 122. And microRNA 122 is an interesting microRNA because it actually promotes HCV RNA accumulation, which is sort of opposite to what microRNAs usually do. And the site 1-bound microRNA actually binds across stem loop 1 of the HCV genome, and this is predicted to stabilize stem loop 1. And as I told you, PCBP has been shown to bind to stem loop 1. So the proximity of these microRNAs to this poly-RC binding protein actually prompted me to think about whether maybe miR-122 and poly-RC binding protein 2 are physically interacting, and if they're helping each other to mediate this effect on viral RNA accumulation. And when I did some IPs, I found that microRNA 122 actually indeed interacted physically with poly-RC binding protein 2, both in infected and uninfected liver cells. So we hypothesized that PCBP proteins bind to the HCV genome. They also bind to the microRNA 122, and that these interactions are facilitating HCV RNA replication by circularizing the HCV genome. And so we wanted to really characterize and identify the binding sites for poly-RC binding protein in the HCV genome, and then elucidate whether these are forming a functional complex in the viral life cycle. So how do we identify the PCB binding sites in the HCV genome? So we used a pretty new approach and pretty powerful approach to study protein-RNA interaction. It's called iClip. It's individual nucleotide resolution, cross-linking immunoprecipitation. So essentially what we do is we cross-link the cells, and this causes a cross-link between the protein and the RNA. And then we sort of fragment the RNA, and we pull down, we pull down the poly-RC binding protein and whatever RNA is stuck to it. And then we essentially sequence that RNA, but because we've introduced this cross-link site where because we've introduced this cross-link site in the protein, we can actually identify the specific nucleotide that the protein is interacting with. And so this is a really powerful tool to study protein-RNA interactions because it gives us so much information, and it gives us the exact nucleotide that it's binding to. And because the cross-linking can be performed on the cells prior to cell lysis, it really gives us an advantage because it gives us the unperturbed environment of this protein-RNA interaction in the cell. So we performed PCBP iClip analysis on HCV infected and uninfected cells, and this is just some data from Cellular transcripts that I'm showing you. So we are on par with previous studies. We had about 23.5 million reads. And most of our transcripts are about 40 nucleotides in length. And as you'll see on the far right, we actually found that over 90% of the transcripts were protein coding genes. So this is consistent with known poly-RC binding protein activity. And most of the activity, most of the binding sites were in the 3' UTR of genes. And interestingly, the sequences that were enriched in either the 5' UTR, the coding sequence, or the 3' UTR were actually different depending on the location of where the poly-RC binding protein was, although they were all enriched in poly-C or poly-U sequences. And something that actually jumped out at us is actually the sequences in the coding region actually were in-frame proline residues, so 3 proline residues in frame that were enriched. So this might suggest novel roles about PCBP activity when binding in the in the coding sequence, because it might have something to do with ribosome processing or co-translational folding of proteins. So what about an HCV? So we actually did this in 2 different genotypes of HCV because we figured that things that are important to the viral genome, binding sites that are important to the virus life cycle, will be present in more than one genotype. And so we compared PCBP binding sites between JFH1 and the H77 genomes. And we were able to identify 6 distinct binding regions in the HCV genome that were conserved between the H77 and JFH1. And to our surprise and to our sort of excitement, we found that the PCBP reads actually mapped, 4 of the 6 of them mapped to structural motifs in the viral genome that had annotated roles in the HCV life cycle. So as I said before, we saw binding to stem loop 1, which was known in the literature, so we sort of confirmed that, which was great. And we also saw binding to the poly-U, poly-UC region, which is consistent with PCBP activity. But we also saw an interaction with stem loop 4 of the viral iris, and this is where the start codon is of the viral RNA. And we saw binding to a region that's involved in a kissing loop interaction important for viral RNA replication, as well as the stop codon. So the next steps that my lab is going to pursue are really what are the roles of PCBPs in the HCV life cycle. So we'd like to do some mutational analyses on these sites, and then do some PCBP depletion and overexpression studies, as well as expressing PCB mutants that are defective in either their protein-protein interactions or in RNA binding activities. And then we'd like to assay for roles in translation, replication, genome circularization, and packaging. We also want to know what host or viral factors are involved in this complex and are helping mediate these interactions, and whether the PCBPs and the miR-122, since they're in such close proximity and they physically interact, if they're coordinating to regulate viral RNA accumulation. So in summary, we know that the PCBP proteins are major RNA binding proteins in the cell. They have roles in RNA processing, in translational control, and RNA stability. And they participate in the life cycles of numerous viruses, including hepatitis C virus. Our iClip analysis revealed that PCBP binding sites were in numerous cellular transcripts, and the majority of them were protein-coding genes, and primarily in the 3' UTRs. And they were enriched in poly-C or poly-U sequences. And the enriched sequences differed between the 5' UTR, the coding sequence, and the 3' UTR, suggesting that there may be location-specific roles for the poly-RC binding proteins in mediating their effects. And finally, this is sort of our model for PCBP interactions in the HCV life cycle. So we know that PCBPs interact with stem loop 1 and sequences that are important for both translation and replication. So it might be mediating circularization events or the switch from translation to replication. It also interacts with microRNA 122, and so might be helping mediate the effects on viral RNA accumulation. So with that, I'd really like to thank Howard Chang at Stanford University. He's my collaborator on this project, and Lance and Ryan, who really helped me uptake the iClip stuff and helped me do all the bioinformatics for this. And then on the right side is my lab that I've just started a few months ago. So it's Annie, Anissa, Jacob, Kevin, and Sean. And I'd like to thank everyone for giving me the opportunity to speak and my funding sources. So thank you so much.
We have time for 1 or 2 questions to Selena. Marcus?
Thank you. Great talk.
Thanks.
Very interesting. I might have missed it, but what happens when you knock down PCB in the fully infectious HCV system or
yeah, so you start to lose viral RNA and proteins if you knock down poly-RC binding proteins. Based on the literature, we haven't explored that as much yet in my lab, so I only have preliminary results. But it seems that we're not really sure whether it's affecting translation or replication based on previous results thus far.
Hi, Selena. Nice talk. It's binding a lot of sequences, if I understand correctly.
Yeah.
So either it binds multiple sequences.
Oh, yeah.
Or it's binding non-specifically to any RNA. So.
Yeah. So actually, the sequences are enriched in poly-U, poly-C, and the protein actually has 3 RNA binding domains. So it could be that it's sort of grabbing to like the stem loop 1 and the iris site at once, or it could be mediated through protein-protein interactions because it's known to dimerize as well. Generally, all of our sequence areas that popped up are enriched in a triplet of C or U, because that's what fits in the KH domain binding pocket. Yeah. So I think that's where we'll sort of focus on in our mutational analyses.
Okay. Does it have a lot of positively charged amino acids? Because it might. I'm just concerned it might be just sticky.
Yeah. Well, I think, so basically based on previous analyses of where they did pull downs, then they did microarrays to see where PCBP proteins were binding, we're finding the same genes that they found. So we identified 97% of the genes that were identified in a previous study. So I don't think it's just sticky.
Last question, Joyce.
Okay. Did your iClip studies, that was done in the context of fully infected cells?
Yeah, so the H77 was a deletion mutant that had a deletion between the E1 and P7 protein, but JFH1 was infectious virus.
Yeah,
and
so you haven't looked at
in
it blocking translation or blocking replication, that sort of thing? You haven't done that yet?
No, I haven't done that yet.
Okay.
I'm going to sneak in a question. Any interactions with the negative strand?
No, 0, actually. I was wondering if we would see something with the negative strand, but we had no binding at all to the negative strand. None of our reads mapped there.
So a nice control.
Thank you.
Thanks a lot, Selena.